Hi everyone, this is Dr. Santosh. Today we'll talk about human genome. We'll learn about ourselves, our genome. So here you can see the human genome is made up of 3200 mega bases, okay, million bases. So this much bases are present in haploid set of chromosome. That is 23 chromosome has about this much bases. And then in that bases you can divide that base, those all bases into two groups. The bases which contain gene and the other part is intergene, DNA. So DNA which is present between the gene. Now you can use the earth as an analogy to understand the genome. So you can see in earth, you know that most of the part of the earth is water, which is not suitable for our living. We cannot live there. And only less than 1/3 of the earth is land. That land too, not all lands are habitable. There are only a part of land which is really habitable. Other part of land there is desert, there may be something in Antarctica, there is too much of cold desert, so we cannot live on all the lands. There are only some lands, some part of land where we can live. The same thing here you can find in human genome. The human genome, the most of the human genome, 1/2 of the human genome, it does not code for any protein. They do not code for any protein. Only 1/3 DNA have code for protein. In that 1/3 also, you can see there most of the part there are pseudo genes, gene fragments, and untranslated intronic regions are there. The regions that is present between the gene fragments. Okay, the real gene fragments. 20 to 25,000 genes that has been expected in humans. That those genes are located on 48 million bases region only. So if you see the whole in human genome, you will find that only 1.5% of the DNA codes for the gene. Now 98.5% of DNA does not code for gene. Now you'll think, what are they junk? No, they are not junk. Most of the region when you see, especially the 65% intergenic region, you'll find repetitive sequence and regulatory sequence and repetitive sequence. So 15% of the DNA they are repetitive sequences, and then 50% of the whole human genome is repetitive sequence, and these repetitive sequences are called microsatellite, mini satellite, satellite, and transposons. I'll be doing lectures, separate lectures on this microsatellites, mini satellites, and those transposons. But just understand that these repetitive sequences are unique in different persons. So we take this repetitive sequence to understand the individuality of a person. The technique called DNA fingerprinting is based on the sequence of this repetitive sequences. Now regulatory sequences, we have the region for microRNAs and other regulatory regions. For example, the regions where protein will go and bind and will regulate the genes. So these sequences, regulatory regions, is 15%. So if you see the whole genome is 65% non-intergenic and 35% is genic region. In that genic region also, there's only 1.5% is the gene. Other are pseudogenes, gene fragments. And it is interesting to note that as the individual is evolved, you'll find evolved living beings will have more intergenic region than non-coding region. So because human is the most evolved being, that is why you can find the most of the genome of human, they are non-coded, non-protein coded. And remember, at last only 1.5%, that is 48 megabase, around 50 megabase of genome is only protein coding, and the number of gene that this bases 48 megabase contain is 20 to 25,000. Thank you.